Alright, so let's understand what the scientist has discovered from scratch. They made tiny beings, or discovered tiny beings. But what microbiologists would say is these are contaminations, but a scientist is, on the other hand, would say that these are not contaminations. So, by Kate Hutt, what she wrote was scientists say that they have believed to build a cell from scratch. For the first time, they can feed, grow, replicate like a natural cell. So, this can be a contamination from a microbiologist's perspective, right? But no, this is not. This is a breakthrough in synthetic biology. So, yeah, this is a biological that has been made synthetic. This could usher an era made to other organisms that function like living machines. So yeah, organisms are exactly like living machines if it is being synthetic biologist. So biological. So so Kate Adamala, a synthetic biologist and a professor at a University of Minnesota, and her team constructed the cell piece by piece from non-living chemical components. This creation is a limited and a fragile prototype. So it is a prototype. It is not yet lived for hundred of days, for many days at all. So and does it replicate or not? Let's see. The creation is a limited fragile prototype, but it could help scientists better understand the origin of life and could potentially be programmed to help mitigate some of the world's biggest biological problems. The cell is non-specific, neither plant nor animal. Non-specific meaning it has not yet been discovered of what type of the cell is, because many of the organs are not visible here. As you can see in the picture, this is just a cell wall or a membrane even, and I don't know if there is a nucleus or not. It is not very clear. So I know the full ingredient list of the cell. We all know that, right? The biomolecules, the nitrogen, carbon, oxygen, and other. I know exactly what chemicals, what molecules, at what concentrations, she said. And it is fully defined, which means we can engineer it. She is just saying that we can create a cell, we can create any type of cell, but not any type. It can become of any type. We just need those sorts of elements: carbon, hydrogen, nitrogen, oxygen. I think these are all the elements that we need to make a cell. So scientists have for decades bioengineered natural cells, and the most researched cell is E. coli. E. coli bacteria cells to manufacture insulin. It could manufacture anything actually. So it treats diabetes, and scientists argue synthetic cells are the next frontier. That's why biotechnology is exactly going so bigger. So they could potentially lead to a development of a new cancer treatment and novel ways to capture carbon or manufacture chemicals. Cells are fundamentally building blocks of life. We all heard about that, and I think this we have been heard about it trillions of times. And we have to understand that this cell is not something that a human cell is, or this is not a human cell. So we need to learn about what cell are we actually planning to make, because it could interrupt the natural way of life. The synthetic cell that Adamela and her colleagues built was not life created in a lab. So this is not a scientific person doing a thing in a lab, but a genuine milestone on the road toward that question. An associate professor in biochemical technologies at Imperial College London, Yuval Ilani, who was not involved in the work, that building yourself from a scratch means you are no longer tied to constraints and evolutionary baggage of natural biology. This means that there is no such thing as survivalness. You just need to make up a chemical that arises a life, that just lives on, a chemical that lives on. We have seen many of these chemicals, even crystals, even the odors that is there in ammonia. Everything, the odor lives on, right? So it's actually just making that into a particular living being, a living thing, which many of the microbiologists would say that it's a contamination, but I don't think so. So to my mind, this is a real advance in long running effort. But this is still a prototype. It does not live on as we do, but it still is a new being, a new living being. And it is also a field of synthetic biology that is separate from stem cell research. Stem cell research meaning a cell which is regenerating, which has the total potency to regenerate itself. It could create uh, immune cells, red blood cells, and every blood cells that we all have, every cells that we all have. It's all of the stem cells. That was the source of the stem cells. I think the stem cells is just that type of cell which can regenerate and which can create other types of cells. So an incredibly wimpy organism. Why do we say wimpy? Because we do not know what type of the organisms, organelles are there, but we do know that it's functioning. So we're hoping we're really starting to the true age of bioeconomy. So she's saying, what about bioeconomy? So she's saying that we could make an organism out of scratch. We could make anything out of scratch. And biologically, if I could make you a hen out of scratch, then I would not have to pay for that chicken that I ate. So, enabling technology that will let people engineer biology. So, this is the thing. And they named as a spot cell. I think you have seen the Sputnik of the Ross that is there. So, Adamela described spot cell as an incredibly wimpy organism that right now basically does nothing. I think most people would argue that it does not replicate, it does not have DNA or some kind, but it has a molecules of it. So it does nothing other than eat and occasionally make daughter cells. So it does replicate, it does do a certain kind of division. That means it has a nucleus, nucleolus, uh, the DNA divides. So each generation requires feeding roughly 12 hours to replicate at a temperature 30 degrees Celsius. So it's more like a bacterial cell than any other cell. It is a prokaryotic or even less than that, less developed than that. So E. coli divides every 30 minutes. It is a general form of every bacteria, but not every bacteria. And roughly under 12 hours, there is a couple of organisms in a plate. And here goes the DNA. The synthetic cell's genome is far smaller than that of a natural cell, with 90,000 base pairs. E. coli's has 4.6 million base pairs. Every organism has a little bit of millions of base pairs. While it can replicate like a natural cell, the synthetic cell deploys a different mechanism. So it has a different mechanism. Let's understand that. A natural cell uses cytoskeleton. I think we all have known about the cell division method, the mitosis. These are done from cytoskeleton, the strings that are there, right? Spindle fibers, I think you have heard it, right? So a structural framework of that spot cell lacks. So they don't have a cytoskeleton. Let's understand what do they have. The synthetic cell, by contrast, produces proteins which crowd at the membrane, forcing it to split. So it is like splitting. I think the budding is a good way of understanding this. Yeast cells does these kinds of replication. So it's just the beginning. Spot cell is also unable to make its own ribosome. So what does this mean? This is a prokaryotic, right? But let me ask you one question. Does prokaryotic have ribosome or not? So it will tell us how much you have learned so far. The key parts of the natural cell that makes protein. Instead, it uses E. coli's ribosome that are supplied through feeding. So they are feeding E. coli's ribosome here. We need to understand that the what type of digestion is there in the cell. What does the cell digest? So the cell can digest E. coli's ribosome, if I've heard it right. What does this mean is that they do not make their own proteins. They make proteins by replicating the proteins that they have eaten. I think this sounds more careful. It's just the beginning, Adamela said. It's a cases that we're hoping to build on, and that's a significant. Yeah, it is a significant thing, but because now we actually have some reasonable idea of how to build on it. So we are actually trying to build on something like that. So maybe we can feed it and feed an elephant to become an elephant, but no, it does not work like that. The later on would say that the natural cell is mimicking or the created cell is mimicking as if it's a natural cell, but the mimicking is because of the natural cell that it had been eaten, because there is no other thing that it can eat or digest. So this cell does not exactly mimic a natural cell, even though it wants to. But that is unnecessary a flaw. Some of these lifelike behaviors are achieved by mechanisms quite unlike those used in biology. So this was said by an email, Ilani. This matters because synthetic biology is not always about imitation. Sometimes it allows us to do things differently and to take shortcuts. I don't know what this is saying. So other scientists are not involved in the research, described the work as an exciting as advancement. Now, why the spud cell? I think the spud cell is because it's a new thing from a spud. It's like a scratch from a bee. That's why it's named as a spud. A pile of chemicals and a naturally evolved cell in nature. It is said naturally, but I think it needs to be said as man-madely. Um, so she called the research important and impressive because it gave result. Every research, when it gives a result, when it gives a prototype, then it is said to be impressive. It is said to be important. But if it does not, it just creates a theory, then it is said to be yet to be discovered. So probably the biggest breakthrough in recent times in the synthetic cell field. Yes. So, as you might have heard, that the AI is actually discovering many proteins, and even we can discover many proteins, but we cannot discover many cell organisms, cells, unless we make them. So, making a synthetic cell helps us to understand the exact minimum requirement for our life and how life might have emerged from chemistry, and that's a cool thing to understand. So there is one experiment, or there is one research that has been going on while saying this, because I think the Ellis here, Mr. Ellis here, have known one thing, that if we know how much life, that how much requirements that we need for a life, then we could actually bind it down into 1%. And if 1% is only enough for us to live on, then I think we do not need more livestocks. What I'm saying is that in space they do these kinds of things because there would be a lack of scarcity of food, water. So they would just eat a protein gum, and the protein gum is full of spirulinas and other synthetic things, probiotic things, with lots of moisture on it. It's like a gel and aero nutritions, which, when eaten, you would not get hungry, you would not get dehydrated for a longer period of time. And I think this is a great thing to say. He knows most of the things that we do not know. Many of the scientists knows like that. So this is a fluorescence microscopic, I think, but not identical to the cells that found in nature. So this can be called as an alien cell, but no, a man-made cell. So this is a peer-reviewed journal, and is it life? So that's a very good question about it to ponder upon. The spot cell cannot be said to evolve. What does this mean? A key achievement for their work, the researchers said, was showing that the synthetic cells are subject to the forces of selection, the process by which certain trait becomes more or less common. When they introduced a genetic change that increased potential production of growth proteins, cells carrying it grew and divided faster. However, because this change was introduced to the system rather than arising a spontaneous genetic mutation, its spot cell cannot be said to evolve. So what he's saying is that the gene that we have, every living being has a mutation, right? Every being has a certain millions of genetics, millions of the uh, nucleotides that allows it to become mutation, to evolve. Every mutation is an evolvement of such kind. So what spot cell cannot do is evolve. He's saying that. So to go more deeper, I think we have to understand the proteins that are there in the spot cell, and how they could grow, like how they could divide like a yeast cell, but not like budding. It's not a budding. It's just protein being two. So I think it's just a reaction from protein uh, by protein. So not can spot cell really be considered, nor can spot cell really be considered life. said Indy, an associate professor of bioengineer at Stanford University. So Indy was not involved in the Aromena's research, but a co-founder of Biotic. So we don't totally understand life. Far from it, we don't have an all-powerful ability to manipulate matter like stuff and make stuff. I would say Kate has constructed a cell. I don't think she created a life. Indy said, noting that while physicists still don't fully understand the mysteries of gravity, and so this is just a comment that has been said. So she's saying that I think this is just a constructed cell, not a life. So many of the people, I think we have known that we can construct a cell, but we cannot construct a life. It's not about to build one. It's about the creation that just happens one. So I think this is all about today. The scientists that have made from scratch. I mean, that's a bravo. That's brilliant. I mean, this is what scientists can do. Thank you.